Hello everyone, hope you're doing well. So, till today we have completed the pre-fertilization events in the gymnosperm. Today we're going to discuss about the fertilization as well as the double fertilization events takes place in the gymnosperm flower. In fertilization event, fertilization we normally term over here is a double fertilization because here two types of fusion we can see. First is the syngamy and second one is the triple fusion. As we have seen during the gametogenesis in the male plant, in a male gametogenesis we have seen that in the pollen tube there are two male gametes are present. As the pollen tube enter into the synergid cells, these two male gametes will enter or will get discharged into that synergid cells. Now, as soon as these synergid cells will receive the male gamete, they will start getting disappearing. Now, these male gametes are free within this compartment or within this cell. Now, the very first male gamete that will bind with the egg cell which is present near the synergid cell, will bind and fuse with the egg cell. So this will form a zygote. This process of fusion of male gamete with the egg cell is termed as syngamy. Okay, hope you understand till now. This is the first fusion takes place. The second one is the second male gamete which is already discharged into the cell or into the synergid cell will come into the middle of the cell and will bind with the polar nuclei. As we know, the polar nuclei is having the two nucleus. So as there are two nucleus and one nucleus from the male gamete, they will fuse and hence they will form the triploid formation. That means 3N. N plus 2N, they will form 3N formation. That's why it is called the triple fusion. Here three different nuclei will bind and fuse with each other. Hence it is called the triple fusion. See here, what happened in syngamy, the male gamete I have shown over here and the female gamete, they will bind and form zygote. That is, male gamete is always haploid in nature and female gamete also haploid in nature. So two haploid gametes will fuse and will form the diploid zygote. Okay, in the triple fusion, the second male gamete which enter into the filiform apparatus to the synergid cell, this male gamete will fuse with the polar nuclei. This polar nuclei is 2N in nature, that is diploid, and this one is haploid. This haploid plus diploid that will form the triploid formation, that is 3N. Okay, this formation is called the primary endospermic cell. So this was just about the double fertilization. As the term says, two type of fertilization takes place, first one and the second one, it is called the double fertilization. Here, once the fertilization takes place, the synergid cells which are present at the micropolar end and the antipodal cells will start getting disappear. Okay, now here you can see the structure of the primary endospermic cell. As this male gametes will get entered into this cell, they will start converting. This was embryo before. Now in the embryo, as this male gamete will start getting entered, they will convert into the primary endospermic cell. And the nucleus which is present is called the primary endospermic nucleus. Which nucleus is there? This is the formation because of the triploid fusion, and this is primary endospermic nucleus is triploid in nature. Here you can see the primary endospermic cell. This antipodal cells are shown very small inside. It indicates that it is start getting disappeared. These synergid cells, two cells are also getting disappeared, and this one will increase in size and form the zygote. Okay, so this was all about the double fertilization. After double fertilization, the post-fertilization events takes place. Post means after. Post-fertilization, during the post-fertilization event, as we have seen that during the fertilization, zygote and primary endospermic cells were formed. Now what will happen? This zygote will continuously divide and divide mitotically. Okay, so divide mitotically and form embryo. Whereas the primary endospermic cell, they will start getting divide mitotically and form primary endosperm, or you can say the endosperm. Okay, so we will first discuss about the endosperm. Okay, primary endosperm cell that is repeatedly doing mitosis formation. This due to the mitosis, they will make an endospermic tissue. This endospermic tissue, why it is needed? What is the function of the primary this endospermic tissue? This endospermic tissue will, or endospermic cell, they will have in to the embryo. During the embryogenesis process, they will provide the nutrient. Okay, understand? So main function of this endosperm, that is diploid in nature, and function of the endosperm is to provide nutrient during the embryo formation. Some plants or some seeds, they form have got the endosperm, whereas some does not have the endosperm. The seeds which are having the endosperm are called endospermic seeds. Okay, so we can say the seeds which are having the endosperm are called albuminous seeds. Okay, seeds having endosperm within them are called endospermic seeds. And some seeds which do not contain any endosperm are called non-albuminous seeds. See, after cell division process, when the seeds get matured, sometimes this endospermic tissue will get disappear, because all the endospermic tissue will get utilized. So no more endosperm will present at the seed formation. Those are called the non-albuminous seeds. Whereas in some seeds, still there will be some endospermic tissue or endospermic cells will be present. These endospermic cells, some will get utilized obviously, but there will be presence of some endospermic cells and called the albuminous seeds. Okay, understand two type of seeds, albuminous seeds and non-albuminous seeds. Okay, now we'll see there are two types of endospermic cell formation. First one, that is free nucleated endospermic formation, and second one is a cellular type endosperm. In the pre-nuclear endospermic formation, what will happen? Here I have shown just for the understanding one picture. Nothing is like this. Don't consider this as a real figure. That is not in your textbook. Just for the understanding, I have made over here. See, this is the primary endospermic cell. Okay. Now what will happen? This endospermic nucleus will start getting divided mitotically. As it is dividing mitotically, they will not change the triploid stage. They all will be triploid in nature. The nucleus will be triploid in nature. Now, this primary endospermic cell mitotically divide multiple times and form the multiple nucleus in the primary endospermic cell. This multiple nucleus, these are the multiple nucleus. Okay. So, once this multiple nucleus formation takes place, this a vacuum or vacuole created in the middle or somewhere at the primary endospermic cell. As the vacuole is created, it will push the pre-nucleated endosperm. There are multiple nucleus. They will push them apart towards this wall of the primary endospermic cell. So this vacuole is created. Vacuole pushes the endospermic cell towards the cell wall, and those cells will come at the edge and form the wall formation. Here, this is the final stage. This is the wall formation took place, and after some time, this vacuole will get disappear. How? Some plant, as we just for the example, we can see in the coconut, this vacuole we can see in the middle, and the coconut water we can see is the free nucleated and endospermic formation. Here, nucleus formation only takes place. On the top of the nucleus we cannot see the cell wall formation. Okay, so you can consider in other seed we cannot see this type of formation. We have to see the dissection of the lot many this kind of embryos, then only we can see this kind of formation. But real example visually you can see is the tender coconut. In the tender coconut, what we can see, the water which is present in the tender coconut is a free nucleated form, because on those are the nucleus in the form of the water. Those do not have the cell wall formation. Isn't it interesting? This is quite interesting if you learn by heart, and it will be easy for you to understand. Once you understand, it will be easy for you to write in the exam. Okay, now over here you can see this is the second type of formation, that is a cellular type. It is completely opposed to the free nuclear type. How? As I said, in the free nuclear type, this nucleus formation, this nucleus is multiple times getting divided mitotically and from multinucleated chamber. This is something like this. But in the cellular type, what will happen? Opposite to that, as soon as the cell division takes place, they will form the cell wall around the nucleus. So here you can see they have this nucleus were divided and they have formed the cell wall formation, and gradually this is due to the mitosis, same like that, but the lacking thing in that is no cell wall formation, and over here the cell wall formation takes place. End of the process of the maturation, this will form the segmented endosperm. Segment is a separate part. Okay, the example of this free nucleated formation is normally the grains, maize, rice, wheat, and many other grains. And example of this cellular type is in Datura and Petunia. If you want to see this both type of cellular as well as the free nuclear type of endospermic formation, we can see in the coconut. Okay, at the primary stage, in the beginning, the tender coconut, we say there was a free nuclear type of formation, endospermic formation. This tender coconut, as it gets mature, this endospermic free nuclear will get pushed around due to the vacuole creation. They will get pushed on the periphery. Okay, and as they will push those endospermic cells which were pushed apart, they will again start forming the cellular type of endospermic division. So completely mature coconut, free nucleated endosperm which were present in the tender coconut or in the liquid form, they will get utilized during the germination of the seed. Okay, hope you understand till this. Now we will understand about the embryogenesis. Okay, this was the first part, that was about the primary endosperm cell to endosperm formation. As I discussed, endosperm having the seeds having the endosperm are called the albuminous seeds, and seeds not having the endosperm are called the non-albuminous seeds. Now the second one is the embryogeny. Okay, as we have seen during the double fertilization, the formation of zygote it takes place. Zygote is then divide mitotically, and they will form the multiple types of multiple cells, and these cells will form a proper arrangement and form an embryo. So formation of an embryo is called the embryogeny. What happened during the embryogeny? The zygote which are present that will divide mitotically and form the embryo. During this formation, there will be having the two cell stage. Once the nucleus will get divided, this 2N diploid stage nucleus, then it will get divided and two cell stage will form. That will again divide and 4 cell stage, then 8 cell stage. After that, it will convert it into the globular stage. After the globular stage, they will form the heart-shaped stage, and after that, they will form the torpedo stage, and then the mature embryo. Okay, so there will be a continuous cell division and rearrangement of the cell. They will form the mature embryo. After the mature embryo, there are different types of plants. As the plants are different, like monocotyledonous plants and the dicotyledonous plants, the formation of the embryo is different. Okay, we will try to understand the formation of the embryo in both the monocot and the diploid stage. Okay, now we mark it. You can see over here there are two different types of the seed. This is the dicotyledonous seed and this is the monocotyledonous seed. In the dicotyledonous seed, as you can see, there are two cotyledons over here. These cotyledons are the reserve energy that will be utilized during the seed germination. This cotyledon are two, this side is called the dicotyledonous. On the top of dicotyledonous, there is the presence of plumule from which the shoot will get developed. That's why it is called the shoot apex. This one is the cotyle. Above the level of the cotyledon is called the epi. Epi above the level of the cotyledons is called the epicotyle, whereas the below the level of the epicotyle is called the hypocotyle. Here you can see the radicle. This one over here is a radicle from where the root will start appearing. So it is called the root apex. The root is covered with the root cap that gives the protection to the root. So these are the function and the diagram of the diploid seed. Here you can see the diagram of the monopod seed. This is the scutellum. Scutellum is nothing but it's a cotyledon. Here you can see just one cotyledon. That's why it is called the monocotyledon. This is scutellum, and this one over here is the plumule. Here this plumule is also termed as the shoot apex that is covered with the coleoptile. Coleoptile is nothing but is the kind of sheet that enclosed that covers the shoot apex. Epiblast over here, then here you can see this is the radicle. Radicle is the same, the organ, the part which are present in the monocotyledonous seeds and dicotyledonous seeds are the same. Here root cap and that covers the radicle, and the coleorhiza, that is again the sheath which covers the root cap. Okay, hope you understand the structure of the monocot and dicot seed. Over here now you move further, the formation, the transformation which takes place after the fertilization. That you can see in this, that is accessory part which are present into the flowers, such as the calyx, corolla, stigma, style, and rostrum, that will get wither off. Wither off means fall off. Then ovary will convert into the fruit. Ovary ball that will convert into the pericarp, ovule that convert into the seeds. The interdiments will convert into the seed coat. Okay, some seeds which, some flowers which have got one or more interdiments. So in that, one is the outer interdument and inner interdument. The outer interdument will convert into the testa, whereas the inner interdument will convert into the tegmen. Then the micropyle end, which we see the porous structure which is present into the embryo, that micropyle will remain as it is, from where the seed will get air and the humidity or the water. That will be remaining same. So it will be no conversion of micropyle in any other thing. That micropyle will be a micropyle in the seed. This funicle will convert it into the stalk of the seed. Okay. Now there is an important term which is given in the NCERT, that is apomixis and parthenocarpy. In apomixis, the seed will form without fertilization. Okay, so just remember the term and how it is formed. So apomixis, that means the formation of the seed without fertilization, whereas the parthenocarpy is formation of fruit without fertilization. Parthenocarpy, commercially it is very important, because by understanding and by knowing this feature of some plants, farmers or researchers can create, the botanists are creating fruits without seeds. So parthenocarpy is nothing but the creation of fruit without fertilization, and those are very valuable. Third one is polyembryonic. If more than one embryo are present, then it is called the polyembryonic. So this was all about the chapter number 2, sexual reproduction in flowering plant. Okay, hope you understand. Try to understand and prepare the chapter. If you take more interest in that, it will be very easy for you. Thank you.